So now we have a discussion about new frontiers in medical research, therapy, and drug discovery. Today we have Alan Coleman from Harvard University speaking to us from near Bordeaux in France. Welcome.
Okay, thank you very much. I'd like to thank the the organizers for inviting me, and I'm really sorry I can't be with you today, and I hope you are all well. I'm at home giving this talk, and I've sent the wife out, but I can't control the cats, so I hope there won't be any interruption. I'm going to be speaking about pluripotent stem cells in medicine and drug discovery and giving you an update on progress. So just to introduce the main players in this talk, we have the pluripotent stem cells here. I think you can see at the top left, embryonic stem cells, and they're made by dissecting out the inner cell mass of an early mammalian embryo. These cells can be cultured indefinitely, and they have a property known as pluripotency because they're able, given suitable incubation conditions, to differentiate into all the somatic cells that are present in the adult form. Below them, we have another type of pluripotent stem cell called an induced pluripotent stem cell, which has a completely different origin. This stem cell, which has the same properties as the embryonic stem cell, comes from taking somatic cells from a person or a mammal and a mammalian animal and subjecting it to transformation by adding a cocktail of transcription factor genes. Again, it's pluripotent and can give rise to all the somatic cells of the body. And sorry, if I could just go back. Oh, sorry. There are a number of uses one could see these cells having. I'll be going through the first four in this talk. The fifth one, the cultured meat, I'll be speaking about this afternoon in the food tech section of the meeting. So what's the history of these pluripotent stem cells? Well, it started in 1981 with Martin Evans in London producing mouse embryonic stem cells. This was followed shortly after by Jamie Thompson using discarded embryos, I believe, that came from Israel to make human embryonic stem cells. And that in 2006, the first induced pluripotential stem cell was introduced by Shinya Yamanaka. This was in mouse, and that was followed in 2007 by the production of human iPS cells by several groups. So there was a lot of enthusiasm right at the beginning, and a lot of money went into researching the use of these cells. It was thought these would revolutionize medical practice and medical research. What I want to do now is see if any of this promise has been delivered or whether it was all too much hype. So the uses I'm going to discuss are on the left, in vitro disease models. And here we have, just let me get rid of, sorry. I've got all these things on the stuff. The provision of normal and disease cell types not normally accessible or available from volunteers or patients. These can be modeled in various formats. Then there's drug screening, development and safety testing. Normal and disease-affected cell types for drug screening and safety studies can be provided by differentiating these pluripotential stem cells. And finally, cell replacement therapy, where you can look to the provision of healthy cell replacements for defective cells in various disease conditions. Patients can receive cells made from allogeneic sources or even their own autologous sources if it's iPSC. You can take a skin cell from a patient and convert it in principle into iPS cells and then change it to the type of cells you want for cell replacement therapy. So I'm now going to talk about the use of the HPSCs in disease modeling. So why develop disease models using these type of cells? Well, often there's no suitable animal model to study the human disease in, and the use of patient-derived human cells of interest is limited by the availability and numbers of the cells that could be taken from the patient. One could circumvent this in principle with immortalization, but that often changes the phenotype of the cell, which can then not be used. Now, patient-specific iPSC can be obtained capturing monogenic and complex familial diseases, as well as complex diseases of unknown genotypic origin. So if you're talking about iPSC, you could take an Alzheimer's patient where you've got no idea of the genetic cause of the disease and still capture that disease in the form of an iPS without knowing what genes are responsible. These cells can be indefinitely renewed in culture and, as I've said, differentiate into many different cell types. And hundreds of diseases have been modeled using these type of cells. So one of the first, in fact, the first disease model that was done was Leishmanian disease, and this was done in Jerusalem by Nissim Benveniste's group at the Hebrew University. They used PSCs to monitor this particular disease. They followed this study in 2007 with a study of Fragile X syndrome using embryonic stem cells. This is another disease that cannot be modeled in other mammals. So Fragile X is the most common form of inherited mental retardation. It's caused by an expansion of the CGG triplets in the FMR1 gene with 5 to 55 triplets being formed in normal people, but in affected people, they have close to 200 repeats or more in their genome, and this disrupts the expression of this particular gene. And what Nissim's group did was they made embryonic stem cells from an afflicted embryo, which had been diagnosed using prenatal genetic diagnosis and found to have over 170 triplet copies per cell. Surprisingly, the mutant ES cells express the gene. The adult cells don't express the gene in people with this disease, but the embryonic stem cells did express the gene, but this expression ceased during differentiation in the test tube, and it was found to be due to hypermethylation of the triplets. So the model allowed a dissection of the sequence of events by which this repression occurs at the very earliest stages of development, which were inaccessible to any other form of study at the time. Now, the disease modeling can be improved by the use of organoids. Whilst PSC-derived cell types can and continue to be used in 2D culture, these models have disadvantages because they often only contain one type of cell and so lack the cellular diversity as well as proper cell-cell and cell matrix interactions that you would want in a model. However, when you aggregate these cells in low attachment conditions and give them cocktails of particular growth factors, you can differentiate them into little organoids. They self-organize to form miniature organ-like structures which capture several of the heterogeneous cellular and functional properties of the desired organ type. And the cartoon on the graphic on the right shows the workflow. You take an iPS cell from a patient, you'd make one from the patient, and then you'd use directed differentiation to make diseased organoids, which could then be used in drug screening. And the next slide just shows on the left, and I don't expect you to look at this table, which has many examples of diseases that have been modeled using organoids. And this is just one of 5 pages in this paper that I've cited at the bottom. And I just wanted to say that let's get that out of the way, even so, challenges remain, even with organoids, because there's phenotypic variation amongst the sister PSC lines that you make from a given patient. You always want to make several lines to do the experimentation. And the cells generally made from pluripotent stem cells in general have embryonic or fetal cell phenotypes, not adult. So that's a limitation at the moment, which has been worked hard on. And also in the organoids, you have inadequate cellular heterogeneity and tissue patterning. So you might have quite a lot of different cells, but you don't have enough different cells, and they're not arranged as they would be in the tissue. And of course, the final thing is sometimes there's a failure to eradicate the memory of the cell that once was. So you take a skin cell, and some of the memory of being a skin cell remains even when they're in the pluripotent state. But there are ways of circumventing that, and organoids are a really important source of tissue for disease modeling. Next. All right. So uses of PSCs in drug discovery. Sorry, I was having a bit of trouble changing slide there. So I'm going to give you one example, which is ALS, amyotrophic lateral sclerosis. It's a heterogeneous neurodegenerative disorder leading to progressive loss of upper and lower motor neurons. We call it in the UK motor neuron disease. And basically 90 to 95% of this disease appears in a sporadic form with no known mutation at the moment. The last 5 to 10% are familial, with a number of genes implicated, and some of them are shown here, SOD1 and the others. Now, Hideyuki Okana's group made iPSC-derived motor neurons from a whole bunch of sporadic cases, familial cases, SOD1, FUS, TDP43, and healthy donors. And they made motor neurons, which they then screened with a library of approved drugs that is on the FDA website. There's 1,232 drugs which have been approved for other indications, and they screened their motor neurons looking at 7 different phenotypic traits that change in ALS, and they identified a potential drug shown on the right, ropinirole, and which was already available for another indication, and then that was subjected to a phase 1/2A clinical trial, which is shown on the left here in the graphic, but it goes through what I've just described in terms of making the cells. The summary of the trial was that it was safe to use with these patients, well tolerated. They were able to show that lipid peroxide in the cerebrospinal fluid is a potential biomarker to assess disease progression, and that the mode of action of this drug in this particular indication was it worked by inhibiting one of the pathways of cholesterol biosynthesis. Next, want to talk about the use of the PSCs in drug safety and toxicology studies, and this is where you look at prescribed medicines before you actually release them, before they're approved for prescription, and look at their cardiotoxicity. And a number of medicines have been found which disrupt that normal rhythm of the heart and lead to a condition known as torsade de pointe, which can lead to death, sudden cardiac death. And you can see the profile of the ECG underneath. Sorry, just get the mouse here. And the FDA, along with foreign counterparts and pharma companies, other stakeholders, has been looking for a thorough test of cardiotoxicity. And at the moment, PSC-derived cardiomyocytes are thought to provide one of the tests that are going to be included in a mandatory test panel to be used on all new drugs. So now, finally, the final use I wanted to discuss in this short talk is the use of pluripotent stem cells in cell replacement therapy. So in the last 10 years, there have been over 100 clinical trials started using pluripotent stem cell-derived cells in cell replacement therapy. There are 4 types of pluripotent stem cell listed there, but only 2 of them, the embryonic stem cells and the iPSC are actually the ones that contribute to most of these cases, and they're the only ones I'm going to talk about. But there's been 44 different target indications covered, and the next slide just shows the type of cells that have been used for replacement in the last 10 years. And we have retinal pigmented epithelial cells at the top of the list, and you can see the total number shown here, 102 clinical trials, and this is the breakdown of the cell types used. But I only want to give you one example, which is very topical at the moment, which is cell replacement for type 1 diabetes. And here we have patients that have lost their beta cells that produce insulin because of autoimmune rejection. They receive daily insulation jabs. In year 2000, James Shapiro showed that cadaver-source beta cells in the form of pancreatic islets could partially or totally restore insulin independence. In some patients, they had to use immunosuppressive drugs to prevent rejection, and the islets were injected into the patient livers via the hepatic portal vein. Now, in 2014, Doug Melton in Harvard was able to show that they could make these beautiful beta cells, and they went into a clinical trial. This is my penultimate slide, which was taken on by Vertex, and they were able to show that with 6 patients, they all started making insulin from the cells. 2 patients, after 1 year, are completely insulin independent, and the trial is continuing and will be superseded by another trial using encapsulation, and that's starting this year. And my final slide is that PSC disease modeling is flourishing, and whilst there are remaining challenges, it's really providing a lot of information on disease formation for people. The jury remains out on the use of PSC models to identify new or repurposed drugs because no drug has yet been approved, although many are in early clinical trials. For drug safety trials, PSC-derived cardiomyocytes could provide standard tests, and many promising developments, but no cell replacement products have yet been approved, but there are many trials, as you can see, underway, and expectations remain high. Thank you very much.